Welcome to the 8th unit of grade 10, Characteristics of Organisms. In this lesson, we are going to discuss about the basic characteristics of living organisms and the importance of these characteristics for the maintenance of life. So, here we are going to discuss about 8 major characteristics of organisms. Out of these 8, some organisms may show only one or few living characteristics. There may be organisms who are exhibiting all these living characteristics. The common characteristics are shared here. You can see them on your screen. First thing is cellular organization, nutrition, respiration, irritability and coordination, excretion, movement, reproduction, growth and development. So we'll move on to these characteristics one by one. First thing is cellular organization. So you have learned about the structure of the cell as the basic structural and functional unit of life. So there are both unicellular organisms as well as multicellular organisms. In unicellular organisms, the cellular organization cannot be seen. They show organelle level organization. When it comes to multicellular organisms, they exhibit this cellular level organization. So here you are shown some of the unicellular organisms like Chlamydomonas, which is an algae. It is a green alga. And then Euglena, which is a protozoan. Then amoeba, again a protozoan. It does not have a distinct shape. Here in Chlamydomonas, this is the shape and it consists of 2 flagella and there is a cup-shaped chloroplast. And here Euglena, it also consists of a flagellum for locomotion. Amoeba does not have a distinct shape. And again Paramecium, which is a ciliated protozoan, it has the shape of a sole of a shoe and the whole body is covered by cilia. These are used for locomotion. So these are some of the unicellular organisms, and you know bacteria are also unicellular, like Lactobacillus, Vibrio. Likewise, you can name so many bacteria. Then these organisms show, these unicellular organisms show cellular level organization. Sorry, they exhibit organelle level organization. Then we'll move on to multicellular organisms. First we'll learn how the origin of multicellular organisms take place. So we'll take the example of development of embryo of human. So here you can see the human gametes. The male gamete is the sperm and it has a haploid genetic composition, and the female gamete, which we call as the ovum, again it has a haploid genetic composition. Then these two gametes will fuse during fertilization to form the diploid zygote, which is a single cell. So this is how the life of this multicellular organism, the human being, takes place. So though we have several millions of cells in our body, once we are grown up, our life begins with a single cell which we call as the zygote. Then the zygote undergoes cell division. Actually, here onwards, all these cell divisions are mitotic cell divisions, or what we call as mitosis. As a result of cell division, the number of cells increase, and we call this cluster of cells as the morula, and the number of cells will be further increased due to mitosis, and it will form a structure known as the embryo, and the embryo will develop into the fetus. Actually, we call the embryo as the fetus after 11 weeks of development. Then this fetus is developed into an infant, and after the infant is grown up, we call it as an adult. So this is the development of the embryo of human. So this can be taken as an example to show the origin of multicellular organisms. Then we compare unicellular organisms and multicellular organisms. There we can say unicellular organisms have an organelle level organization. There are several organelles inside a cell which are responsible for different functions. So unicellular organisms have organelle level organization, and these organelles are well developed to perform different functions. For example, the chloroplast is responsible for photosynthesis, mitochondrion is responsible for cellular respiration, nucleus is responsible for controlling cellular activities and transmission of genetic material from generation to generation. Likewise, there are organelles which are well developed to perform different functions. Then when it comes to multicellular organisms, we can say tissue and system level organization is there. We'll learn about this tissue and system level organization in a coming example. Then here we can say organs are well developed to perform different functions. Here organelles are developed to perform different functions. In multicellular organisms, organs are developed to perform different functions. If we take the plant body, the leaves, flowers, fruits, these are the organs of a plant. If we take the animal body, heart, liver, kidney, lungs, these are considered to be the organs. Then we'll move on to this diagram which shows the parts of the plant body. So here, there are two main systems in a plant. Above the ground level, we find the shoot system, and below the ground level, we find the root system. So root system consists of, as this is a dicot, there is a taproot as well as there are lateral roots. And these roots consist of root hair cells. And when we consider the shoot system, there are leaves, the stem, flowers, flower buds and flowers. So if we consider the stem and the veins of leaves, they consist of the conducting tissues like xylem and phloem, and the basic units of these xylem and phloem tissues are xylem cell and phloem cell. We learn in grade 11 that there are 4 types of cells in each of these, in xylem as well as in phloem. And here they have shown a cross section of the leaf. Here the epidermal cells are shown, palisade parenchyma cells, spongy parenchyma. Then here the stomatal pore is shown, and they have shown how the stomatal pore is made. There are 2 guard cells. In between the 2 guard cells, there is the stoma or the stomatal opening. So this is how the plant body is made out of cells, tissues and systems. So I must say that in grade 11 first unit you will learn about these tissues and these cross sections and all about these. Then The organizational levels identified in living organisms, we can give examples for these. Then we'll come to the human body. There again you are shown uh, here I have mentioned different systems in the human body: blood circulatory system, digestive system, excretory system, respiratory system, skeletal system, reproductive system, endocrine system, and nervous system. So if we consider the organs present in those, blood circulatory system consists of the heart, and blood vessels are there, and there are several tissues. The heart consists of cardiac muscles, and in the blood tissue there are blood cells, white blood cells, red blood cells. And if we take the nervous system, the basic unit in the nervous system is the nerve cell, or what we call as the neuron. And the skeletal system consists of bones, and there are muscles also, uh, which help for the movements in the body. And in the reproductive system there are gamete cells, and the whole body is covered with the skin, which is an um, epithelial tissue, and this consists of skin cells or the epithelial cells. Likewise, there is a cellular level, tissue level, organ level, and system level organization in human body as well. So we'll name the organizational levels identified in living organisms like this. So these are the organizational levels. A cell, tissue. Cell is the basic unit. A number of cells get together to form a tissue. A number of tissues will form an organ, and a number of organs will form a system, and the collection of different systems will make an organism. So for example, I have taken a cardiac muscle cell. It forms the cardiac muscle tissue, and these tissues are helpful for the formation of the heart, which is an organ. And heart is in the blood circulatory system, and a number of systems like blood circulatory system and all the other systems I mentioned before form the human body, the human being, which is an organism. So you can see examples for each level of organization: cell, tissue, organ, systems, and organism. If we take the plant body, I have given an example: parenchyma cell. A number of parenchyma cells form parenchyma tissue, and this parenchyma tissue is available in leaves as well as fruits and flowers also. Here I have considered the leaf as the organ, and a number of leaves helpful for the formation of the shoot system. Not only the leaf, shoot system is the system which consists of organs including leaves. Then shoot system and root system collectively form the plant body. So these are the examples for the organizational levels identified in living organisms. So we have discussed the first living characteristic, that is cellular organization. Then we'll move on to the second characteristic, that is nutrition. So nutrition is the process by which organisms obtain materials for the maintenance of life. So if we don't take food, we don't get energy. But taking food is the process of nutrition, that is the process by which we obtain materials for the maintenance of life. So these materials will then undergo cellular respiration in order to produce energy, which is our next topic. So depending on the ability of producing food, organisms are divided into autotrophs and heterotrophs. Here you are asked to define the terms. Autotrophs are the organisms which can produce their own food. Heterotrophs are the organisms which cannot produce their own food. So, animals are heterotrophs. Autotrophs are then further divided into photoautotrophs and chemoautotrophs. So, photoautotrophs are the organisms which make use of light energy in the production of food. In other words, photoautotrophs are the organisms which make use of light energy in the process of producing food. Examples: green plants, algae, and cyanobacteria. Then chemoautotrophs. Somebody will call these as chemoautotrophs. So here these organisms make use of the energy released in a chemical reaction for the production of food. Example, most bacteria. They make use of the energy released in a chemical reaction for the production of food. Here light energy, here energy released in a chemical reaction. Then photosynthesis. What is this photosynthesis? So this is the word equation which describes photosynthetic reaction. Their major raw materials are carbon dioxide and water, and this happens only in the presence of actually we don't need to mention sunlight here. If we say light energy, it is okay. It is the source of energy and chlorophyll is the substrate. So the factors necessary for photosynthesis are carbon dioxide, water, sunlight, and chlorophyll. When these four factors are present, green plants or the photoautotrophs will produce glucose as the main product, and they will release oxygen as a byproduct. So here the balanced chemical equation for photosynthesis is shown. And this is the balanced chemical equation. Later you will learn how to balance an equation. Here 6 carbon dioxide molecules which are in gaseous state, 6 water molecules which are in liquid state. In the presence of light energy and chlorophyll, they will produce glucose. You know, this is the chemical formula of glucose, C6H12O6, and it is in the solid state. And this is the byproduct, oxygen O2, and 6 oxygen molecules are there in order to balance the equation. 6CO2 plus 6H2O in the presence of light energy and chlorophyll, C6H12O6 and 6O2. These are the word equation and the balanced chemical equation for photosynthesis reaction. So you know that photosynthesis takes place in green plants, and main photosynthetic structures are the leaves. So as a result of photosynthesis, glucose is produced, and that glucose is stored temporarily in stems. This stored glucose is temporarily stored in leaves in the form of starch, and that starch should be translocated to the other parts of the plant, that is in the form of a sucrose solution. Stored starch in the leaves are translocated to the other parts of the plant in the form of a sucrose solution. Then after that, again that is stored as starch in other plant parts such as stems, roots, and fruits. So that is all about photosynthesis. And if you think of a food chain as well as a food web, you will be able to uh, think of the autotrophs and the heterotrophs who are existing in the environment. So this is what we have discussed. And photosynthesis happens mainly in leaves, and I explained what happens to produced glucose after photosynthesis. Then these are the different modes of nutrition shown by the animals and plants as well as microorganisms. So you know about autotrophs, then there are heterotrophs, all the animals are heterotrophs. And in addition to that, there are decomposers. Decomposers are mainly of two types, detritivores and saprophytes. So these detritivores are depending on dead or decaying organic matter, and saprophytes are feeding upon nutrients obtained from decaying organic matter. So detritivores, for example, we can take the earthworm as a detritivore, and bacteria and fungi, who are the main decomposers, come under saprophytes. And another mode of feeding or another mode of nutrition is parasitism. Parasites are living on a host, and they obtain nutrients from the host's body. So far we have discussed about cellular organization and nutrition. Then we'll move on to respiration, the process by which we obtain energy. Now you learned during nutrition we obtain the materials required to sustain life. During respiration, what happens is the food that we have taken undergoes digestion, and end products of food digestion or the digestive end products undergo cellular respiration to produce energy. Digestive end products undergo cellular respiration to produce energy. Cellular respiration is a series of biochemical reactions. So, you know that cellular respiration takes place inside the mitochondrion. So, if we talk about respiration, cellular respiration is a part of it. So, muscular movements that allow the passage of air in and out of respiratory organs are called respiratory movements. So, later in grade 11 you will learn that the external part of the respiratory system, the external respiration, and the internal respiratory movements, and finally the cellular respiration taking place. So here you are given a simple activity to show that carbon dioxide is released during respiration. This is the setup used for that. If I explain the setup first, materials required are lime water, you know, that is calcium hydroxide, potassium hydroxide, water, 5 equal bottles with corks, certain glass tubes, and a frog. So you can see the 5 glass bottles are connected by this using the glass tubes, and you can see in bottle A there is potassium hydroxide, and this tube is immersed in potassium hydroxide. The second tube is out of potassium hydroxide, it is in air. Then you can see it is connected to bottle B, and this end of the tube is again immersed in lime water or calcium hydroxide. The end of the next tube is out of calcium hydroxide, and the end of the tube is close to the frog inside bottle A, and the next tube, and the end is immersed in lime water again, and this end is out of lime water, and then this end is immersed in water, and you can see bottle E is connected to a suction pump or a tap. So once we have put the things as shown in the diagram, we have to open the tap. That will allow a continuous flow of air from P onwards. Likewise, a continuous flow of air will be maintained once we open the tap. So here we have to use colorless lime water in both instances, and you know potassium hydroxide is important to absorb carbon dioxide gas. And you know here we are going to show that carbon dioxide is released during respiration. The frog is used as a living organism for that purpose. If we can't find a frog, we can use germinating seeds like germinating green gram or something. For bottle C, arrange the apparatus as shown above and remove water in bottle E by opening the tap. Then we'll move on to the questions based on this. What is the purpose of adding KOH into A? So potassium hydroxide is added into A in order to absorb carbon dioxide gas which is coming from outside. Normal air enters from P, and carbon dioxide in that air is absorbed by potassium hydroxide. Then we know that there is no carbon dioxide in the air which is passing from bottle A to B. Then what can be observed in lime water in B and D? Give reasons. So since there is no carbon dioxide in the air which is passing from bottle A to B, you know lime water will not turn milky. You know carbon dioxide gas is absorbed, is obtained by lime water, and that lime water turns milky if there is carbon dioxide. In B, lime water does not turn milky as there is no carbon dioxide. If I mention the reason, if you can remember in grade 6 also you have learned that colorless lime water is used to identify carbon dioxide gas. Once carbon dioxide gas reacts with colorless lime water or calcium hydroxide, it forms calcium carbonate and water. As calcium carbonate particles get suspended, this turns milky color. So our observations are in B, lime water does not turn milky. In D, lime water turns milky. The reason is during the respiration of the frog, carbon dioxide gas is released, and that gas gets dissolved in lime water in D. Therefore, in D, lime water turns milky. Then why E is connected to a suction pump? I told you it is to maintain a continuous flow of air. What can be inserted in C instead of the frog? So you can mention germinating seeds like germinating green gram. Then another activity to show that oxygen is absorbed during respiration. During respiration, oxygen gas is absorbed and carbon dioxide gas is released. So this is the experiment to show that oxygen is absorbed for respiration. You need two conical flasks. These are conical flasks. Then glass tubes are needed. Then a rubber tube, small test tube, two beakers, colored water, and germinating seeds. So you can see both these A and B setups are similar except the tube with potassium hydroxide in setup A. So you can understand here you are said to close the flask with the cork lid connected to a rubber tube, and a U-tube is attached as shown in the diagram. This is the U-tube, U-shaped tube. After some time, tighten the rubber tube. So this was opened earlier to external air. After some time, this was tightened to stop the flow of air from outside to in. Then here what happens is that here we assume that the carbon dioxide released and oxygen gained is more or less the same. The two volumes are the same. That is an assumption. So here in A what happens is during respiration carbon dioxide gas is given out and that gas is absorbed by potassium hydroxide, and oxygen gas is obtained for the respiration by the germinating seeds. So two gases are removed from this volume inside the conical flask. As gases are matter, to fill that gap, this colored water go up through the U-tube to occupy that space. But in B, oxygen gas is obtained and carbon dioxide gas is released. As we have assumed also, the volume of the two gases are the same. There is no space for this colored water to climb up or to rise along the U-tube. Therefore, colored water level in tube B or setup B does not change, but in A it will be changed. So we'll move on to the questions based on this activity. What are the assumptions made here? So you can say all carbon dioxide is absorbed by potassium hydroxide. That is one assumption we made. All carbon dioxide gas is absorbed by potassium hydroxide. And the second assumption is that the volume of oxygen obtained and the volume of carbon dioxide released are the same. Then explain the observations as I have explained. In A, as carbon dioxide gas is absorbed by potassium hydroxide, there is more space that is empty, and oxygen is also taken by the seeds for respiration. Therefore, there is much more space empty. To occupy that space, this colored water rise along the U-tube. But in B, as there is no space to occupy, colored water does not rise up. Then what is the drawback of this experiment? Actually, the drawback in this experiment is we can't prove that the gas which is taken or obtained for respiration is oxygen. We can't prove that it is oxygen, but we know that oxygen is available about 21 or 20% in the atmosphere. So that is, if we consider this height as simple h, so h over 5, so 20% means 1 over 5. Actually we can't prove it like that. So that is the drawback in this experiment, that we can't prove the obtained gas is oxygen. From this activity we can't prove it, but we can design another activity to show that. Then the next living characteristic is irritability and coordination. To understand those two terms, here I have given a simple representation. So there are a lot of changes happening in both internal and external environment. Those changes that take place which are strong enough to bring about a response in us are known as stimuli. Singular is stimulus. Change in internal or external environment which is strong enough to bring about a response is known as a stimulus. So once there is a change, or once we obtain a stimulus, actually that is obtained by the receptors in our body. There are 5 receptors, or what we call as sensory organs. These are the organs which detect stimuli. The organs which detect stimuli are known as receptors or the sensory organs. If we mention the 5 receptors, ears, eyes, nose, tongue and the skin, these are the 5 sensory organs we have. Then stimuli are taken by those receptors, and then finally we have to come up with a response. If I mention an example, your hand touches a hot kettle accidentally. So the stimulus or the change in the environment is heat. So that is received by your skin, by the skin of your hand. So the receptor or the sensory organ is the skin. Then ultimately your response is you take off your hand quickly. Taking off the hand is the response. To give out that response, an effector becomes helpful. Remember, the effectors are always either glands or muscles. Here in my example, the effector is the muscles of your hand. And once we get a stimulus, until we come up with a response, the coordination part is done by several systems in the body. It may be either the nervous system or the endocrine system. Then so we have defined stimuli and receptors. So here you can mention the names of receptors. So if we take ears, the stimuli is sound, eyes, stimuli, light, skin, there are several stimuli, uh, vibrations, heat, pressure, touch, likewise, pain. Then if we take the nose, smell, tongue, it is the taste. Then a response is a reaction according to the change in the environment, or a reaction according to the stimulus obtained is known as a response. Then irritability is the ability to respond to stimuli. So the characteristic of living organisms is irritability and coordination. So irritability is the ability to respond to stimuli. Coordination is the communication between different organs during responding to a stimulus. Communication between different organs during responding to a stimulus is known as coordination. So the effectors are either muscles or glands, and they are helpful to come up with a response. Then you are asked to give examples separately for the instances where plants and animals respond to stimuli. So I mentioned an example where animals respond to stimuli. So some animals are running away when they hear a sound. when we touch a hot object, we take off our hand immediately. Once we step on a thorn, we take off our foot immediately. Likewise, then plants also respond to stimuli. The stimuli may be touch, light intensity, change in light intensity. For example, leaves of mimosa fold when they are touched. Then leaves of thor, tamarind, sesbania or katurumurunga fold at night because of the less light intensity. So these are the instances where plants respond to stimuli. And also, plants grow, the tip of the plant or the shoot tip, shoot apex grow towards light. That is also a response given out by plants. So both plants and animals have irritability and coordination. Then the fifth living characteristic, that is excretion. So excretion is the removal of byproducts from the body that are produced during metabolism. Removal of byproducts from the body that are produced during metabolism is known as excretion. Then what are these metabolic activities? Actually, we can say metabolic activities are the sum of chemical and physiological activities within the cell, and these are for your extra knowledge. Metabolic activities are of two types, that those are anabolism and catabolism. During anabolism, simple compounds get together to form a complex compound. Their energy is stored in the complex compound. During catabolism, a complex compound is broken into simple substances. Their energy is released. So these are metabolic activities. So excretion is the removal of byproducts from the body that are produced during those metabolic activities happening inside living bodies. So here this table is write about the excretory organs present in human body and the form of excretion as well as the excretory materials. If we take lungs, it is also an excretory organ. Form of excretion from lungs is exhaled air, and then the excretory materials from lungs are carbon dioxide and water vapor, carbon dioxide gas and water vapor. Then if we take the skin as the next excretory organ, the form of excretion is sweat, and the excretory materials which contain in sweat are mainly water and salt. In addition to that, there may be a certain amount of urea as well. Main excretory materials in sweat are water and salt. Then the main excretory organ, that is the kidney. So the form of excretion is urine, and the excretory materials are urea, water and salt. Water and salts. There are different types of salts. So kidney is the nitrogenous, kidney is the organ in which nitrogenous excretion of human body mainly takes place. Then plants also excrete, do you think? So during respiration of plants, carbon dioxide is released as an unnecessary substance. Therefore, carbon dioxide becomes an excretory product during respiration of plants. Then, during photosynthesis, plants release oxygen as a byproduct. Their oxygen becomes an excretory product by plants during photosynthesis. So both this carbon dioxide and oxygen are released through stomata in leaves as well as the lenticels. Then we'll move on to the next living characteristic, that is movement. So both plants and animals move. Why do they move? They move as a response to stimuli. Now you know what the meaning of the term stimuli, the changes in the environment. So sometimes whole organism moves. So when the whole organism moves, we call it as a locomotion. Locomotion, this happens in animals, and sometimes a part of the organism moves. When we take unicellular organisms, they use flagella, cilia as well as pseudopodia for locomotion. So if you can remember, at the very beginning I showed you flagella are present in Chlamydomonas, Euglena, and cilia are present in uh, Paramecium, and pseudopodia they are in amoeba. They use these for locomotion. The whole cell moves. And in unicellular organisms, organelles also show movements. The internal organelles also show certain movements. When we come to multicellular organisms, whole body or part of the body moves with the help of muscles. If you think of your body, sometimes the whole body moves and sometimes the part of the body moves with the help of muscles. When we take plants, plants also show certain movements. As I mentioned earlier, shoot apex moves or grows towards light. We call it as a positive phototropic movement or positive phototropism. You have learned about these in grade 9. Then the root apex grows or moves towards gravity, that is the ground. Therefore, we call this as positive geotropism or negative phototropism. But there are certain exceptions like pneumatophores of mangrove plants. They show negative geotropism because these roots come up from soil in search of air. Therefore, these pneumatophores show negative geotropism. And later you will learn what happens to pollen grains once they are deposited on the stigma of a flower in a coming unit. There the pollen tube grows towards the ovary for fertilization to take place. So there we call this type of a movement as a positive chemotropism. And in order to show these movements, you learn that these movements take place as a response to stimuli. So there are different stimuli like light, darkness, chemicals, gravitational force, heat or temperature, as well as vibrations or touch. So as a response to those stimuli, both plants and animals come up with movements. Right. Then the seventh characteristic is reproduction. You know, reproduction is the production of new organisms or new generation by a unicellular or multicellular organism for the continuation of their species. Production of a new generation by unicellular or multicellular organisms for the continuation of their species is known as reproduction. So this reproduction process can be divided into two main types as sexual reproduction and asexual reproduction. So during sexual reproduction, fusion of gametes of two individuals of the same species take place. The gametes of two individuals of the same species takes place. There are two individuals, male individual and the female. So the gametes will fuse during sexual reproduction. So in a coming unit, you have to learn about this sexual reproduction separately. Then during asexual reproduction, single organism produces identical offsprings. That means there is only one individual. It will produce identical offsprings are the members of the new generation. The new individuals produced by this single organism are identical. That means they are more or less similar to each other. As examples, uh, plants undergo vegetative propagation. That means, for example, propagation of new plants by vegetative structures of plants. For example, if we plant a twig of a plant, it will produce an identical offspring or identical plant to the mother plant. Then budding takes place in hydra, and fragmentation takes place in planaria. That means the body is broken into several fragments. Then regeneration that happens in echinodermates like starfish. That is about reproduction. I am not going to explain it further because there is a separate lesson for this reproduction process. Then the last characteristic is growth and development. So what is this growth actually? Growth of a unicellular organism can be defined as the increase in size and the volume of the cell. Growth of a unicellular organism can be defined as the increase in size and the volume of a cell. The size and the volume of the cell increases, then we can call it as a growth of a unicellular organism. Examples for unicellular organisms: paramecium, yeast, chlamydomonas. Then growth of a multicellular organism is, this occurs by means of mitotic cell divisions. If you can remember, at the very beginning I mentioned, I took the example of the development of human development of the human embryo. There I mentioned that the zygote, the single cell, divides, or it undergoes mitotic cell divisions, and it forms a number of cells, a cluster of cells, which we call as the morula. Then the morula develops into an embryo, and then into fetus, then into an infant and an adult. Likewise, the whole process, the zygote develops into the complete multicellular organism by means of mitotic cell divisions. So growth of a multicellular organism can be defined as the irreversible increase of dry mass, because once a cell absorbs water, its mass increases. But when we take the dry mass, it is the real growth, and it is an irreversible process. Once an organism is grown, really we can't take it to the initial status again. If you think of yourself, you have grown, you have undergone the growth as an irreversible process. You can't be that small child again. So remember, growth is the irreversible increase of dry mass. Then what is this development? That is the increase in the complexity of the cell. That means, now if we think of our body, we are not that the cluster of cells which we saw at the very beginning. Our body has been differentiated into different systems. Before that, there are different tissues, organs. So this cluster has been differentiated, modified, adapted for different functions and different structures. The same cluster has been differentiated into form different tissues, different organs and different systems. That is what we call as the differentiation, increase in the complexity of the cell. Increase in the complexity of the cell is known as the development. So this growth and development includes three steps. These are irreversible increase in size of the cell, that is the growth, increase in number of cells by cell division, the number of cells is being increased, and these cells are differentiated into different organs and systems. Irreversible increase in size of the cell, increase in the number of cells, and cell differentiation. These are the steps in growth and development of an organism. Then, this oxonometer is a setup which can be used to show the growth of a plant. Again, this is not that practical, but you have to learn about this setup as a setup used to observe the growth of a plant. So here you can see there is a potted plant and the shoot apex is connected with a string, and the other end of the string is connected with a weight, and it is sent across a pulley, and the pulley is hanging here, and the pulley is connected to an indicator. So when the pulley is rotating, the indicator will also move. And here there is a scale which is kept on a stand. So when the indicator moves, we can get the reading out of the scale. So here what happens is when the plant grows, the height of the shoot apex increases. So because of the weight, the string will be pulled downwards like this. So the pulley will rotate once the weight is moving down. So as a result, the indicator will also show a slight movement, and you will be able to obtain the value. So this is used to measure the growth of a plant. This is known as the oxonometer. According to the figure, the thread is connected to the shoot apex of a potted plant and it is sent through a pulley and a weight is hung onto it. You are asked to observe how the indicator moves. White thread, right. That is about the growth and development. Then we have discussed about all the 8 factors that we mentioned at the beginning: cellular organization, nutrition, respiration, irritability and coordination, excretion, movement, reproduction, then finally growth and development. And after that, there are certain things for your knowledge. White thread-like mass on a pile of decomposing garbage is the somatic part. Somatic means the growth part of a fungus. So we have observed the white thread-like masses on a decaying log, on garbage, spoiled food. We have seen. So that is, these white thread-like structures are the fungal hyphae or the growth part of fungi. Later, some of these are transformed into mushrooms, which are the sexual reproductive structures of fungi. Mushrooms are sexual reproductive structures of fungi. Actually, these information are given in order to give you an idea that there are visual living structures in the environment, and some of these living structures cannot be identified actually as living things. So to give you an idea about this, these information are given. You can observe lichen growing on a coconut trunk, or ferns and orchids grow on a wall, mealybugs on chili or papaw plants, mealybugs, which are white in color again, small fragile white eggs on leaflets of Sesbania caturumurunga, and identify whether they are living or non-living. These are given as examples. Once you see lichens, you have seen definitely on large trees there are white or ash or sometimes light green color structures, patches like structures on those large trees. These are lichens. Um, Actually, a lichen is a symbiotic relationship in between fungi and algae, or this may be a relationship in between fungi and cyanobacteria, fungi and algae, or fungi and cyanobacteria. They make a symbiotic relationship in order to form lichens. Then sometimes living or non-living nature of some substances cannot be identified easily. Living or non-living nature of some substances cannot be identified easily. If you look at those uh, different colored masses on tree trunks, you will think that whether they are living or non-living. The mealybugs, they don't actually move sometimes, so once you see them you will think whether they are living or non-living. The fragile white eggs on leaflets of Sesbania, it is difficult to identify whether they are living or non-living. Bacteria cells can be dried into a powder, and you have seen yeast, which is a fungus, and it is used to make bread, which is available in the market. Yeast is available in the market as a dried powder. So there are instances where we think whether these are living or non-living. Then these days we are talking about virus a lot. There are a lot of viruses. So we don't consider that viruses are living substances, and we don't consider them as non-living substances because they have characteristics in between living and non-living. So if we consider virus, some living entities cannot be easily identified whether they are living or non-living. Example, virus. Actually, viruses are very small and they can be observed only through the electron microscope, and they are about um, one of 1,000 parts of the size of a bacteria. That means if a virus is very small, a bacterium is 1,000 times larger than a virus. A bacterium is 1,000 times larger than a virus. Viruses are that much small. So they show living or non-living features according to their status. Therefore, we don't call them as living organisms, and we can't say that they are non-living as well. If we consider the structure, this is the structure of a model of a virus, which is seen through the electron microscope. So they have a DNA or RNA molecule, DNA or RNA, both do not exist, which is covered with a protein capsid, and there is a tail. Here the tail fibers are also shown. A virus is not considered as a cell. They are composed of a nucleic acid enclosed by a protein capsid. The nucleic acid may be either DNA or RNA. Viruses with different shapes and sizes have been identified. If you have time later, you can search the images of virus. There are so many nice shapes, but they cause a lot of harmful diseases both to human beings, animals, as well as plants. Viruses do not possess any organelles for metabolic reactions. So these facts are very important because you have to remember these for your MCQs. virus becomes active only inside a host cell. So when a virus is on the external environment or on a non-living surface, it cannot be activated. It becomes active only inside the host cell. So once the virus is fallen on a living cell, then it will release the genetic material, either DNA or RNA. Then it starts to multiply inside the living cell. That is how viral diseases become fatal. You are given some examples for common diseases in plants caused by viruses: banana bunchy top disease, curly leaf disease of chili, and tobacco mosaic virus, which causes disease to tobacco plants. And there are so many animal viral diseases: dengue, influenza, common cold, AIDS, mumps, chickenpox, Ebola, rubella, chikungunya, hepatitis, SARS, which is severe acute respiratory syndrome, polio, and the well-known COVID-19 or the coronavirus. Actually, COVID-19 is coming under corona family. And you know the long term of this acquired immunodeficiency syndrome, AIDS, which is caused by HIV, human immunodeficiency virus. So, I think you have gained a clear idea about the characteristics of living organisms. So, after studying this, you can do the exercises uh, which are coming under Unit 8 in your textbook.